Dear students, in this class, what we are going to see is the factors affecting microbial pathogenicity. What it means is, when do we call a microorganism as a pathogen? When it causes disease, right? So what are the characters of a microorganism which makes it pathogenic, which makes it to sustain its life in our body and cause damage to our body? So this is what we are going to see. What are the various characters that is present in that organism, which we call here as factors responsible for microbial pathogenicity. Whenever pathogenicity is being studied, these two terms you will always come across, pathogenicity and virulence. So are they same? Are they different? This is what we are going to see here. Basically, pathogenicity is defined as the ability of a microorganism to cause disease or to cause tissue injury. So this here it is either present or absent. So it is basically a qualitative term. A microorganism will be a pathogen or it will be a non-pathogen. Pathogen means it can cause the disease. Non-pathogen means it cannot cause the disease. So this is what pathogenicity talks about. Whereas what is virulence? Virulence is the severity of the disease caused by a pathogen. It talks about specifically about the pathogen, and it talks specifically about the degree of pathogenicity. That means a pathogen can be can cause a very mild type of disease, or it can cause a disease which is very severe in nature. So it could be a mildly virulent pathogen or it could be a highly virulent pathogen. Sometimes among the pathogens itself there will be some strains which cannot cause the disease. So such strains are called avirulent. So basically virulence is a quantitative term. That means we are going to measure the pathogenicity of an organism. If the pathogenicity of the organism is very less, then we call that organism as a mildly virulent organism. Whereas if the pathogenicity of the organism is very high, very severe, then we call that organism as a highly virulent organism. Now look at this chart carefully. This is the life cycle of a pathogen. The pathogen is first going to enter inside the body through some portal of entry. It could be any route, skin or respiratory tract or gastrointestinal tract or genitourinary tract. Through some route entry point, it is going to enter inside the body. As soon as it enters, the first step it does is it is going to attach to our body cell, on the surface of our body cell. So that is called adhesion. Once it gets attached on the body cell, the next thing it does is it is going to penetrate inside. It is going to penetrate, get spreaded from one cell to another cell, and it is also going to get spreaded from one tissue to another tissue, and it is going to penetrate inside the body. It will try to penetrate inside the body. So that is called invasion. Once it invades the body tissues, the next thing it does is it will increase in its number by using the body mechanisms, by using the body fluids and enzymes and all other things, nutrients. It is going to increase in its number. That is called multiplication. And when it multiplies, at the same time it will also release different types of enzymes and toxins which is going to damage the tissues that is present in the body. So a particular tissue will be affected, so that is called infection of the target. And once the tissue is damaged, you are going to see the symptoms in our body, and that is when we call it as disease. Now, once it damages the tissue, starts damaging the tissue, the next thing it will do is, it will, from that human body, it will try to get spreaded to another human body. So it is going to come out of the human body through some portal of exit. So this is basically the life cycle of a pathogen inside one particular human body. So once the organism, the pathogen, enters inside the human body, from that moment till it comes out, it has to face a lot of problems inside the human body, because the human body is not going to keep quiet. It has its own resistance mechanisms to fight against the pathogen. So if the pathogen is causing the disease, then it must be having its own mechanisms to get rid of all these difficulties and cause the disease. So right from the point it attaches itself to the human body cells, and then getting spreaded inside the tissues and multiplying inside the body cells, and then targeting a specific tissue. So to do all this, it must have certain specific mechanisms, and these specific mechanisms are going to be called virulence factors. So what are the various virulence factors a pathogen will be having to cause the disease? So these are some of it that I have mentioned here. The first one is adherence factor, which is necessary for getting attached on the body cells, that is adhesion. Second is invasiveness, that is the ability of the organism to spread from one cell to another cell, from one tissue to another tissue. So that is invasion. Next is it is going to release certain toxins and enzymes which is going to damage a specific tissue in our body, and then it will also have certain mechanisms to evade our resistance mechanism, because as soon as the pathogen enters, the body is going to fight against it using a specific type of cells called phagocytes, and the pathogen must be having some mechanism to escape these phagocytes. So those are called anti-phagocytic factors. And then there is another very important character that we need to remember, that is antigenic variation, and finally iron requirements. So we are going to see these factors a little more in detail. The first virulence factor that we are going to see is adherence factor. Adherence or adhesion means attachment of bacteria to tissue surfaces after it enters inside the human body. Now, how this attachment is very specific in nature. In fact, that means it just not go and bind on any body cell as such. There must be surface receptors on host cells, and there must be adhesive structures on the bacterial cell surface. So host cell andre body cells ಮೇಲೆ surface receptors ಇರಬೇಕು, bacterial surface ಮೇಲೆ adhesive structures ಇರಬೇಕು, which we usually called ligand. So there must be an interaction taking place between the ligand and the surface receptor, and after this interaction there will be a specific binding taking place between these two cells. So that is how the pathogen will attach itself on the host cell. Now for this binding to take place, attachment to take place, as I said, there must be some adhesive structures present on the pathogen surface. So what are these adhesive structures? They are usually called adhesins, and the usual adhesins what we have studied till now is like this: fimbriae, pili, capsule, slime layer, teichoic acid. All these structures you have studied in detail in your first semester itself. The external structure of bacterial cell wall ಅಂತ ಪಾಠ ಮಾಡಿದಿರುವಾಗ ಇದನ್ನೆಲ್ಲ ಹೇಳಿರ್ತಾರೆ. The fimbriae, and then pili, capsule, the outermost slimy layer that you will find in some of the bacterial cells, mainly the pathogenic ones, and then slime layer, glycocalyx layer, or teichoic acid, the cell wall component present in the bacterial cell wall. So all these are external cell structures of a bacterial cell, and one or two of them will be responsible for binding on the surface of the host cell. And what is this? Spike proteins. These are usually present in viral cell surface. Viral pathogens alli ee spike proteins irutte. Coronavirus allu kuda spike proteins ಬಗ್ಗೆ ಕೇಳಿರ್ತೀರಾ. So go through that in detail. Fine. So these are some of the adhesins that you will find on the surface of the pathogens which help them to bind on the host cells. And most of these adhesins are proteinaceous in nature, and they are also antigenic in nature. Now what is this antigenic? Immunology alli idanna tumba detail agi ಮಾಡ್ತಾರೆ. I will just give you an idea here. Antigenic means when these structures are encountered by our immune cells are going to produce antibodies against these structures. Antibodies against fimbriae or pili or capsule or spike proteins will be produced inside the host cells, and these antibodies will be very specific to these adhesins. So such structures which will stimulate antibody production in the body are called antigens. So all these adhesins are usually antigenic in nature. That means when they enter inside our body is going to produce antibodies against them. Fine. Now, this is one way of attachment of the pathogen on the body cells. There is another way also, which is the character cell surface hydrophobicity. Most of the pathogens will show this character, cell surface hydrophobicity, when they enter inside the human body. What is the cell surface hydrophobicity? See, hydrophobicity means repulsion against water or any liquid substance, right? Hydrophobicity. So when the pathogen get repelled by the liquid material inside the body, then they have a very good capacity to go and bind tightly on the surface of the body cells. See, ಏನಾಗುತ್ತೆ ಅಂತಂದ್ರೆ ಈ pathogens ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ ಎಂಟ್ರಿ ಆಗುವಾಗ they will be initially present in the mucosal secretions. ಅದು oral ಆಗಿ ಇರಲಿ ಅಥವಾ nose ಆಗಿರಲಿ, when they enter inside, initially they will be present in mucosal secretions. That means liquid substance ಅಲ್ಲಿ suspend ಆಗಿರುತ್ತೆ. But ಹಾಗೇನೆ ಇದ್ದು ಬಿಟ್ರೆ they can never bind on the surface of the body cells. So body cells ಅಲ್ಲಿ bind ಆಗಬೇಕು ಅಂತಾದ್ರೆ they have to get repelled from the liquid secretions. Liquid secretions ಇಂದ ಹೊರಗಡೆ ಬಂದು ಹೋಗಿ tissue ಮೇಲೆ bind ಆಗಬೇಕು. So if it has to be repelled, then it should have the character of hydrophobicity. So this is what we call cell surface hydrophobicity. ಎಷ್ಟಕ್ಕಷ್ಟು ಈ ಹೈಡ್ರೋಫೋಬಿಸಿಟಿ ಪ್ಯಾಥೋಜನ್ ಅಲ್ಲಿ ಇರುತ್ತೋ ಅಷ್ಟಕ್ಕಷ್ಟು ಇಟ್ ಕ್ಯಾನ್ ಗೋ ಬೆಟರ್ ಅಟ್ಯಾಚ್ ಇಟ್ ಸೆಲ್ಫ್ ಆನ್ ದ ಸರ್ಫೇಸ್. ಸೊ ಮೋರ್ ಹೈಡ್ರೋಫೋಬಿಕ್, ಬೆಟರ್ ಅಡಿಯರೆನ್ಸ್ ಆನ್ ದ ಸರ್ಫೇಸ್ ಆಫ್ ದಿ mucosal linings. Okay, so these two characters are going to help the pathogens in their adherence on the surface of the body cells. So this is just a diagrammatic representation of how attachment or adhesion happens. So using the fimbriae, which is present on the surface of the bacterial cell, they can go bind on the surface of the tissue, or using capsules that surrounds some of the bacterial pathogens, they can go and bind on the surface of the host cell, or the spike proteins, as I said, which is present on the surface of the viral capsid. So these spike proteins, they can go and specifically bind on the surface of certain cell receptors, and these receptors are present on the host cells. So again, there is a ligand receptor binding happens between the host cell and the pathogen, by means of which it goes and attaches itself on the surface of the specific host cells. This is just extra information that I would like to give here. Not all the pathogens will have same type of adhesins or same type of adhesion characters. They use different mechanisms to attach themselves to the host cells. So I have just mentioned a few pathogens here, the disease that they cause and how they, what type of adhesion mechanism they have. For example, if you take Neisseria gonorrhea, they have fimbriae by means of which they go and attach on the genital epithelium. Whereas if you see Vibrio, they have glycocalyx anchors which can attach itself to the intestinal epithelium. Whereas if you see some spirochetes like Treponema, which causes syphilis, they have hooks. Since their body is spiral shaped, they have tapered hooks with which they go and attach themselves on the host cell. Whereas if you take viruses like influenza virus, they have spikes on their surface with which they attach themselves on the host cell. So different organisms, pathogens have different ways of going and attaching on the body cell surface. The next virulence factor we are going to see is invasiveness. What is invasiveness? The ability of the pathogen to penetrate and spread in the host tissue after establishing infection. That means after it gets itself attached on the host cell surface, how it penetrates and spreads inside the host tissue. So this ability is called invasiveness. So a pathogen may be less invasive or highly invasive. A highly invasive pathogen cause generalized infection, whereas a less invasive pathogen cause localized infection. What does that mean? See, if the pathogen cannot get itself spreaded very well, then it is going to be constrained in one particular area alone, and it will cause damage in that particular region only. So in that case, a less invasive pathogen, it is a less invasive pathogen, and it will cause a localized infection. For example, Staphylococcus abscess. So this is a boil which is seen in one particular area on the skin. So this is a localized infection. Why? Because the pathogen cannot get spreaded very well throughout the body. Whereas if we talk about highly invasive pathogens, they will get spreaded throughout the body very effectively. So they are going to cause damage in many parts of the body, which will result in generalized infection. For example, streptococcal septicemia. Here the organism is going to enter the blood, and through that circulation it can reach different parts of the body. So you will start seeing symptoms in many parts. So that is when we call it as a generalized infection. Now, when we talk about invasiveness, we say that the organism gets spreaded. Just by means of multiplication and spreading, can the organism cause damage? No. Many a times the tissue damage happens through two different ways. One is the organism, the pathogen is going to release some kind of a damaging enzyme, or it is going to release some kind of a toxin. So what are these enzymes and toxins? That is what we are going to see in detail in the next slide. Now, what are toxins? These are toxic products produced by some pathogens when they are multiplying inside the body tissues. They are not produced by all pathogens. Only some pathogens produce these toxic products called toxins. Now, there are two types of toxins which we come across. One is the exotoxins, the other one is the endotoxins. First, we will see what are the exotoxins that we commonly see, and some examples too. Exotoxins are toxins secreted into the surrounding medium by the pathogen when it grows. When some pathogens grow, they produce the toxin and release it into the environment. So such toxins are called exotoxins. And these exotoxins are usually heat labile. That is, they will get inactivated when the temperature increases. They are heat labile and they are usually proteinaceous in nature, and they are highly potent. What does that mean? Even a very small amount of toxin is enough to cause very high damage to the body tissues. For example, if you see tetanus toxin, which is released by Clostridium tetani, just 1 milligram of this tetanus toxin can kill more than 1 million guinea pigs. And there is another interesting fact here. That is, 3 kilogram of botulinum toxin can kill whole world population. 3 kg of botulinum toxin. So that is the potency of these toxins. They are very highly potent. They can damage the body tissues in nanogram quantities. Right now, there is another character about exotoxins, that is, they exhibit specific tissue affinities and pharmacological activities. What is meant by this? That is, these toxins, when they are released by the pathogen, they again will go and bind on specific tissues in our body. For example, if we say neurotoxin, the neurotoxins will go and attack mainly the neuron cells, neurons. Whereas if you see nephrotoxin, these toxins will go attach themselves with the kidney cells. Enterotoxin will go get attached themselves with the intestinal epithelial cells. So they have specific tissue affinities, and they also have specific pharmacological activities. That means when the damage is seen, the damages will be very distinct, very unique to that particular toxin. It will not be a general form of damage. Fine. Now that is about tissue affinities and pharmacological activities. And these exotoxins are generally formed by both gram-positive and gram-negative bacteria. One example for exotoxin released by gram-positive bacteria is tetanus toxin, that is released by Clostridium tetani. And example for gram-negative bacterial exotoxin is cholera toxin, which is released by Vibrio cholerae. Fine. And the last point here we need to remember is exotoxins are also highly immunogenic or antigenic. Just like how I told you about adhesins, these exotoxins are also highly antigenic or immunogenic in nature. This is a slide showing you the mechanism of action of diphtheria toxin. Diphtheria toxin again is an exotoxin released by the gram-positive bacteria, Corynebacterium diphtheriae. Fine. So how this toxin acts on the host cell has been depicted in this picture. You see this host cell, that is the human body cell, the plasma membrane. Diphtheria toxin, which is a dimeric exotoxin, will be released by the bacterium. Dimeric in the sense it has two subunits, A subunit and B subunit. With the help of B subunit, this exotoxin will first bind on the surface of the receptor present on the plasma membrane. Now, once the binding takes place, this exotoxin will be taken up inside the host cell with the formation of a vesicle. So, in a coated vesicle, this dimeric exotoxin will enter inside the host cell, and initially the host cell pH will be 7. So, at that time the dimeric exotoxin will be integrated, whereas some reactions take place inside the vesicle which will bring down the pH to 5. So at this acidic pH, the dimeric toxin will get dissociated. So A subunit and B subunit will get dissociated. Now here the A subunit is important because it is going to do the further action. The A subunit will be released outside the vesicle, and it is an enzyme, the A subunit. It is going to inhibit the action of a very important protein here, that is elongation factor 2. I don't want to tell the other details. Just remember this. The A subunit is an enzyme which will inhibit the action of elongation factor 2. And if you remember, elongation factor 2 is a very important factor necessary for protein synthesis. So if its action is inhibited, that means protein synthesis of the cell will be inhibited. So if protein synthesis stops, then the cell will die. Right? So that is how the cell will be damaged by the A subunit, whereas the B subunit will be released outside the cell. Fine. So this is how the toxins will work inside the body to damage the host cells. The next type of toxins are endotoxins. Toxins which are not secreted into the surroundings but remain associated with the pathogens are called endotoxins. They will be present in association with the pathogen when the pathogen is alive. But once the pathogen dies, they will be released from the cell wall. So these endotoxins are usually an integral part of the cell wall of the pathogen. They will be present associated with the cell wall, the external structure of the cell wall. And we know that bacterial cell wall are made up of lipopolysaccharides, and usually these lipopolysaccharides will act as endotoxins. And unlike exotoxins, they are heat stable in nature. That is, they will not be inactivated at a higher temperature, at an elevated temperature. And they are found mainly in gram-negative bacteria. Again, unlike your exotoxins, these are found mainly in gram-negative bacteria, because we know that gram-negative bacterial cell wall is highly complex in nature, unlike gram-positive bacterial cell wall. ಗ್ರಾಮ್ ಪಾಸಿಟಿವ್ ಸೆಲ್ ವಾಲ್ ಅಲ್ಲಿ ನೋಡಿದ್ರೆ ನಿಮಗೆ ಪೆಪ್ಟಿಡೋಗ್ಲೈಕನ್ ಪ್ರಾಮಿನೆಂಟ್ ಕಾಂಪೊನೆಂಟ್ ಆಗಿರುತ್ತೆ. ವೆರಸ್ ಗ್ರಾಮ್ ನೆಗೆಟಿವ್ ಸೆಲ್ ವಾಲ್ ಅಲ್ಲಿ ಪೆಪ್ಟಿಡೋಗ್ಲೈಕನ್ ಇಸ್ ವೆರಿ ಲೆಸ್. ಔಟರ್ ಲೇಯರ್ ಅಂತ ಇನ್ನೊಂದು ಕಾಂಪೊನೆಂಟ್ ಇರುತ್ತೆ ವಿಚ್ ಫಾರ್ಮ್ಸ್ ದಿ ಮೇಜರ್ ಕಾಂಪೊನೆಂಟ್ ಆಫ್ ಗ್ರಾಮ್ ನೆಗೆಟಿವ್ cell wall. So this outer component with all its complex structure can act as a very good endotoxin, and they are poor antigens, and that means they are not highly immunogenic in nature. ಬಾಡಿಯಲ್ಲಿ ಇದ್ರೂ ಕೂಡ ಅದಕ್ಕೆ ಆಂಟಿಬಾಡಿ ಪ್ರೊಡಕ್ಷನ್ ಜಾಸ್ತಿ ಆಗಲ್ಲ. ಸೊ ದೇ ಆರ್ ಪೂರ್ ಆಂಟಿಜನ್ಸ್ ಅಂಡ್ they are active only in large doses, not in smaller doses like your exotoxins. Exotoxins nanogram quantity ಅಲ್ಲಿ ಇದ್ರೆ high damage, heavy damage ಇರುತ್ತೆ ಅಂತ ಹೇಳಿದೆಲ್ಲ. Endotoxins are not like that. Only when they are present in very high quantities, they cause some significant damage to the body cells. And they also do not have any specific pharmacological activities. That is, when they are present, you will see very generalized effects in the body, like elevated temperature, fever irutte body alli, athava coagulation of blood irutte, diarrhea or inflammation or weakness. Ee ತರ ತುಂಬಾ generalized effect irutte body alli because of endotoxins. So these are the characters of endotoxins. So whatever I told you about exotoxins and endotoxins, I have summarized in this chart. Their characters and the difference between the exotoxins and endotoxins. Characters you need to know here is what is their potency or toxicity, whether they are potent at a very low concentration or at a higher concentration, and what type of effects they have in their body, specific effects or generalized effects, and what is their chemical composition usually, whether they are proteins or lipids or polysaccharides, and whether they are heat labile or heat resistant, that character. And why should we talk about this head stable and heat resistant character? The main reason is we would like to develop them as toxoids. ದಟ್ ಇಸ್ ನೀವೇನು ವ್ಯಾಕ್ಸಿನ್ಸ್ ಆಗಿ ಕೊಡ್ತೀರಲ್ಲ, ಟೆಟನಸ್ ಟಾಕ್ಸಾಯ್ಡ್ ಅಂತ ಬರುತ್ತೆ, ಡಿಫ್ತೀರಿಯಾ ಟಾಕ್ಸಾಯ್ಡ್ ಅಂತ ಬರುತ್ತೆ. ಸೊ ಈ ಟಾಕ್ಸೈಡ್ ಪ್ರೊಡಕ್ಷನ್ಗೆ ಈ ಕ್ಯಾರೆಕ್ಟರ್ ನಮಗೆ ಬೇಕು. ಅದರದ್ದು ಕೆಮಿಕಲ್ ಕಾಂಪೋಸಿಷನ್ ಏನು, ಅದರದ್ದು ಹೀಟ್ ಸ್ಟೆಬಿಲಿಟಿ ನೇಚರ್ ಏನು, ಆಲ್ ದೀಸ್ ಆರ್ ನೀಡ್ ಟು ಬಿ ನೋನ್. ಸೊ ಟಾಕ್ಸೈಡ್ ಫಾರ್ಮೇಶನ್ ಕ್ಯಾನ್ ಬಿ ಡನ್ ಆರ್ ನಾಟ್. Usually exotoxins can be converted to toxides, whereas it is not possible with endotoxins. And then how the response is seen in the body. As I said, these are highly immunogenic, whereas these are poorly immunogenic in nature. And fever stimulation, pyrogenicity idea. If you see that with exotoxins it is usually not seen, whereas with endotoxin pyrogenicity is seen. Fever production, fever causation is seen. And whether they are released or not from the pathogenic cell. So that is what has been seen here. They are usually secreted from the live cell, whereas here they are secreted only after the cell dies. And sources, exotoxins are usually released from both gram positive and gram negative organisms, whereas endotoxins are seen only in gram negative bacteria. So the next important virulence factor, which is generally seen in all types of pathogens, are enzymes. See, when we say toxins, toxins are usually seen in bacteria, whereas these enzymes are seen getting released from pathogenic bacteria, pathogenic fungi, protozoa, and even worms. So all these pathogens secrete exo-enzymes that break down and cause damage on our tissues. So I have just mentioned 4 important exo-enzymes here. The first one is mucinase. It digests the protective coating on mucous membrane, and one example for this mucinase is the enzyme released by amoebic organism causing amoebic dysentery, Entamoeba histolytica. It releases this enzyme mucinase, which disrupts the mucous membranes. The next is keratinase enzyme. This digests the main component of skin and hair, that is keratin. So keratinase enzyme is going to hydrolyze keratin. That is how it causes damage to skin and hair. And this is secreted by fungi that cause ringworm disease, dermatophytes and then ಹೇಳ್ತೀವಲ್ಲ. These dermatophytic fungi, they release this keratinase enzyme. The third is collagenase. Collagenase is an enzyme which digests the principal component of connective tissue. We know that connective tissue is there in our body. So the enzyme collagenase released by the pathogen is going to affect the collagen present in the connective tissue. And one example for this is Clostridium species and even certain worms release this collagenase enzyme, and that is how they invade our body tissues very effectively. And the last one is hyaluronidase. This enzyme digests hyaluronic acid. Where is it present? This is the important substance that cements the cells together. The cementing substance anta ಏನು ಹೇಳ್ತೀವಲ್ಲ, in all animal tissues, one cell will be attached with another cell by means of a cementing substance present in between. And one of the important component of this cementing substance is hyaluronic acid. And this hyaluronic acid will be digested by the enzyme hyaluronidase, which is present in the pathogen. So by releasing this enzyme, it will easily digest the cementing substance, and that is how they can easily penetrate the body tissues and enter inside the circulation. So many organisms release this enzyme like Staphylococci, Clostridia, Streptococci and Pneumococci. So just now I told you about the cementing substance present between the cells. So any tissue if you take in our body, this cementing substance will be present, which will keep the cells together, making a proper tissue. But when the pathogen is present here, it is going to release the hyaluronidase enzyme, and these enzymes will easily digest this cementing substance. And once a gap is formed between the two cells, the pathogen can easily penetrate this tissue and reach the next layer. And that is how it will keep on penetrating inside our tissue and get spread throughout the body. The next virulence factor that a pathogen can possess is anti-phagocytic factors. This is the very important character that any pathogen that enters our body and penetrates our body tissues must have, so that it can cause the disease. Anti-phagocytic factors. Now, before we talk about anti-phagocytic factors, we must know what is phagocytosis that takes place inside our body. You might have studied this in your lower classes, but let me just refresh your memory here. This is the process of phagocytosis that happens in our body whenever an antigen enters. Antigen, or you can just say a pathogen. So when a pathogen, here I have shown it as a bacterium, just consider it as any pathogen. So when a pathogen enters inside the human body, the first cell that is going to attack this pathogen is phagocytes. For example, macrophages. Macrophages are phagocytic in nature. What do? They will first engulf this pathogen inside them. For this engulfment to take place, the pathogen first should go and bind on the surface of the phagocytes or the macrophage. So after this binding takes place, the pathogen along with the receptor will be just taken up inside this phagocytic cell, and it will form a vesicle like this. So inside this vesicle you will see the pathogen, which is represented in a red dot. Okay. Now once this vesicle inside which the pathogen is present is formed in the phagocytic cell, we call this vesicle as phagosome. Fine. Now, once the phagosome is formed, next step, what happens is, in the phagocytic cell, there is a specialized type of vesicle which is already present, which is called lysosome. What is this lysosome? This is a vesicle inside which you will see many different types of digestive enzymes. Different types of digestive enzymes will be present. Digestive enzymes in the sense, enzymes which are going to attack any complex structure that is present in the pathogen. Right from cell wall to cell membrane to all the internal structures, proteinase irbodu, lipase irbodu, ribonuclease irbodu, ee tara different types of digestive enzymes ee lysosome alli irutte. But it will be present inside a vesicle. On the protective membrane ಒಳಗಡೆ ಈ digestive enzymes ಇರುತ್ತೆ, and that vesicle is called lysosome. Next, second step ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, once the phagosome is formed, the lysosome will go and get integrated with the phagosome. phagosome ಜೊತೆಗೆ ಈ lysosome ಹೋಗಿ ಅಟ್ಯಾಚ್ ಆಗ್ಬಿಡುತ್ತೆ, ಇಂಟಗ್ರೇಟ್ ಆಗ್ಬಿಡುತ್ತೆ. ಇಂಟಗ್ರೇಟ್ ಆದ ಕೂಡಲೇ you will find a structure called phagolysosome. What happens here is the pathogen will be mixed along with all the digestive enzymes present in the lysosome. Okay, so ಏನಾಗುತ್ತೆ ಈಗ, all these digestive enzymes will start attacking the pathogen, will break it into pieces. ಎಲ್ಲ ಪೀಸ್ ರೈಟ್ ಫ್ರಮ್ ಸೆಲ್ ವಾಲ್, ಸೆಲ್ ಮೆಂಬ್ರೇನ್, DNA, RNA, ಏನೇನ್ ಎನ್ಸೈಮ್ ಏನೇನಿದೆಯೋ ಎಲ್ಲವನ್ನು ಪೀಸ್ ಮಾಡಿಬಿಡುತ್ತೆ, ಸೊ ದಟ್ ದ ಪ್ಯಾಥೋಜನ್ ವಿಲ್ ಡೈ inside the phagolysosome itself. Once this digestion gets completed, finally the phagolysosome structure will go integrate with the outer cell membrane of this phagocyte, and all the digested products will come out of the phagocyte. ಈ ಪ್ರಾಸೆಸ್ಗೆ ನಾವು ಫಾಗೋಸೈಟೋಸಿಸ್ ಅಂತ ಹೇಳ್ತೀವಿ, where the pathogen will be engulfed by the phagocyte, will be destroyed by the phagolysosome, and the digested product will be released out. Okay, so this is done by the different types of phagocytes that is present in our body. This is first line of defense. So if a pathogen has to survive this process, only then if it survives this process it can cause the disease, right? ಯಾಕಂದ್ರೆ ಇದು ಇದ್ದೇ ಇರುತ್ತೆ ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ. ಇದನ್ನ ಹೇಗೆ ಎಸ್ಕೇಪ್ ಮಾಡಬೇಕು ಅಂತ ಗೊತ್ತಾದ್ರೆ ಮಾತ್ರ the pathogen can cause the disease, alva? So such factors are called anti-phagocytic factors. So many pathogens produce factors that protect them from phagocytosis process. ಇದರಲ್ಲಿ ಕೆಲವೊಂದು examples I have mentioned here. First one is capsules. If a pathogen has a capsule on its surface, usually the capsule will prevent the first step itself. First step of phagocytosis ಏನು? Binding of the pathogen on the surface receptors of the phagocyte. ಈ step complete ಆಗಿ inhibit ಆಗುತ್ತೆ ಯಾವಾಗ? Only when the pathogen contains a capsule. So for example, Streptococcus pneumoniae, it contains capsule. Haemophilus influenzae, it is capsulated. So such organisms cannot be easily phagocytosed by the phagocytes. ಮಾಡಕ್ಕಾಗಲ್ಲ. ಇದು one way of escaping from phagocytosis. Second is, some organisms like Staphylococcus, they do not have capsule. So capsule ಇಲ್ಲ ಅಂದ್ರೆ ಏನಾದ್ರೂ ಬೇರೆ ಫ್ಯಾಕ್ಟರ್ ಇರಬೇಕಲ್ಲ, ಬೇರೆ ಕ್ಯಾರೆಕ್ಟರ್ ಇರಬೇಕು phagocytosis ಇಂದ ಎಸ್ಕೇಪ್ ಆಗೋಕೆ. Staphylococcus produces proteins called leucocidins, and these leucocidins, they will destroy the phagocytes even before phagocytosis can happen. So leucocidins direct agi phagocytes mele attack ಮಾಡಿ they will destroy the phagocyte cell itself. So idu another way of escaping the phagocytosis process. Third way is, some organisms, some pathogens like Shigella, they escape from the phagosome before it merges with the lysosome. As I said, second step alli phagosome lysosome jotege fuse ಆಗಬೇಕು, ಸೇರಬೇಕು. So before this fusion takes place, Shigella, which has been engulfed by the phagocytic cell, will be escaping from the phagosome. ಅದಕ್ಕೆ ಅದರದೇ ಆದ mechanism ಇದೆ, how it escapes from the phagosome ಅಂತ. For now that detail is not necessary. If you want, you can search the internet, you will get the information. Just remember this, Shigella will escape from the phagosome even before the lysosome gets fused with the phagosome. This is another way of escaping. Escape agi from the phagocytic cell itself, it will get escaped. Okay, so that is another way. The fourth most important way is, some of the organisms, some pathogens, they will resist the toxic products released into the phagolysosome after fusion occurs. That means, ಕೆಲವೊಂದು pathogen phagolysosome alli idru kuda, digestive enzymes idru kuda, they will not be digested by the digestive enzymes. How? Their cell wall will be so strong, it will be so highly protected, so that even the digestive enzymes cannot attack them. For example, Mycobacterium tuberculosis. If you see the cell wall of this Mycobacterium tuberculosis, it is mainly made up of mycolic acid, a lipid. And this mycolic acid makes it very, very resistant to all types of digestive enzymes present in the phagolysosome. So even in the phagolysosome, they can survive matra alla, they can be transported to other tissues also with the help of this phagocytic cell itself, that we will see in TB specifically. So just remember this, they can survive the phagolysosome enzymes. Okay, and there is another way. Some bacteria like chlamydia, they prevent phagocytosis by preventing the fusion of phagosomes with lysosomes. Some organisms like chlamydia are there. Even when they are present in the phagosome, they have a mechanism by means of which they will not allow this process to take place. Phagosome, lysosome jotege fuse agbekalla, ಆ process ನೇ ಮಾಡಕ್ ಬಿಡಲ್ಲ. So these are some of the ways of escaping the phagocytosis process so that they can establish inside the human body and cause disease. So such factors are called anti-phagocytic factors. So the two more virulence factors which I would like to explain in short are antigenic variation and iron requirement of the pathogen. Antigenic variation means some microbes, they keep changing their antigenic structure so that they will keep on escaping from the immune response that gets stimulated in our body. So antibodies formed against one type of antigenic structure protect the host from the pathogen of that type alone, but not from those with other antigenic types. ಈಗ ಫಸ್ಟ್ ಟೈಮ್ ಬರುವಾಗ ಒಂದು ಆಂಟಿಜನ್ ಇತ್ತು, ಸೊ ಅವರ್ ಬಾಡಿ ವುಡ್ ಹ್ಯಾವ್ ಐಡೆಂಟಿಫೈಡ್ ದೆಮ್, ದೇ ವುಡ್ ಹ್ಯಾವ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ಆಂಟಿಬಾಡೀಸ್ ಅಗೇನ್ಸ್ಟ್ ದೆಮ್. ಬಟ್ ನೆಕ್ಸ್ಟ್ ಟೈಮ್ ಆ ಪ್ಯಾಥೋಜನ್ ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ ಬರುವಾಗ ಆಂಟಿಜನೇ ಬೇರೆ ಆಗಿರುತ್ತೆ. ಸೊ ನೀವು ಫಸ್ಟ್ ಟೈಮ್ ಮಾಡಿರುವ ಆಂಟಿಬಾಡೀಸ್ ಉಪಯೋಗಕ್ಕೆ ಬರಲ್ಲ. So next time again you will have to produce antibodies, different antibodies against that antigen. So this is how some organisms keep changing their antigenic structure so that they can escape the immune response in our body. ಇದಕ್ಕೆ ತುಂಬಾ ಎಕ್ಸಾಂಪಲ್ಸ್ ಇದೆ. ಈವನ್ ಕೊರೋನಾದಲ್ಲಿ ಆಲ್ಸೋ ವಿ ಹ್ಯಾವ್ ಸೀನ್ ಸಮ್ ವೇರಿಯಂಟ್ಸ್ ಕಮಿಂಗ್ ಅಪ್, changes with the spike protein structures, right? Same way HIV you would have heard, then they undergo mutation again and again so that their antigenic structure keeps changing. Common cold virus, every year ನಿಮಗೆ common cold ಬರ್ತದೆ ನೋಡಿ, because once virus entry ಆದ ಕೂಡಲೇ antibody formation ನಿಮ್ಮ ಬಾಡಿಯಲ್ಲಿ ಆಗುತ್ತೆ. ಸೊ ದಟ್ ಮೀನ್ಸ್ ನೆಕ್ಸ್ಟ್ ಟೈಮ್ ನಿಮಗೆ ಬರಬಾರದು ಕೋಲ್ಡ್ ಅಲ್ವಾ, ಬಟ್ 6 months once ನಿಮಗೆ ಬಂದೇ ಬರುತ್ತೆ ಕೋಲ್ಡ್, ಅಥವಾ 1 year once ಬಂದೇ ಬರುತ್ತೆ. ವೈ? ಬಿಕಾಸ್ ನೆಕ್ಸ್ಟ್ ವೆನ್ ಫಸ್ಟ್ ಟೈಮ್ ಇರುವಾಗ ಇರುವ ಆಂಟಿಜೆನಿಕ್ ಸ್ಟ್ರಕ್ಚರ್ ಸೆಕೆಂಡ್ ಟೈಮ್ ಅಲ್ಲಿ ಇರಲ್ಲ, ಚೇಂಜಸ್ ಆಗಿರುತ್ತೆ. ಸೊ ಫಸ್ಟ್ ಟೈಮ್ ನಲ್ಲಿ ಏನ್ ಆಂಟಿಬಾಡೀಸ್ ಫಾರ್ಮ್ ಆಗಿರುತ್ತೋ ಅದು ಸೆಕೆಂಡ್ ಟೈಮ್ಗೆ ಉಪಯೋಗಕ್ಕೆ ಬರಲ್ಲ. So that is what we call antigenic variation. Say that happens with many different pathogens. So just go through. There will be two terms I will mention here. One is called antigenic drift. The other one is called antigenic shift. ಇದು ಏನು ಎರಡು ಅಂತ ಒಂಚೂರು search ಮಾಡಿ, you will get some interesting facts there. Okay. And the last virulence factor is iron requirement. See, one of the important nutrient that is necessary for the survival of this pathogen in our body is iron. This is the most essential nutrient required by the pathogen. But when the pathogen enters inside the body, iron will not be available to them easily, because the iron that is present in our body will be in the bound form. Either in the form of hemoglobin or in the form of myoglobin, they will be present in bound form. ಈ bound form ಅನ್ನ ಈಸಿಯಾಗಿ ತಗೊಳಕ್ಕಾಗಲ್ಲ ಪ್ಯಾಥೋಜನ್ಸ್ಗೆ. ಸೊ ತಗೊಳ್ಬೇಕು ಅಂತಂದ್ರೆ they must have some special mechanism. So what are those special mechanisms? That is what I have mentioned here, some two special ways of getting the iron. One is with the formation of special substance called siderophores. Siderophores are iron chelating structures. They act like magnets. So elli iron ಇದೆಯೋ, they will go specifically bind with that iron. Whatever small iron that is available, they will go specifically bind with that iron so that that iron will be available for the multiplication of the pathogen. So this is one way, formation of siderophores by some pathogens. Second is some organisms, especially those which will enter through the intestinal tract, they will utilize iron from hemin that is present in our gut freely. So these are some ways of getting iron. I have just mentioned two. There are many other ways also by means of which the pathogens can get iron and survive in our body. Only such pathogens can survive, that which can get iron in some way, they can survive in our body. Okay, the last slide. As usual, this is a questionnaire slide. You need to go through some literature and find out what are all these terms mean. First is antigenic shift and antigenic drift. If you want, you can refer literature based on streptococcal diseases. There you will get some information regarding this very much in detail, antigenic shift and antigenic drift. Third is spike proteins. Especially I would like you to go through the spike proteins of coronavirus, since it has been in news nowadays. Just find out natural articles kuda ide. Spike proteins of coronavirus anta hakidre ಅದರ ಹೆಸರು, its specific receptors, how it changes, ಇದೆಲ್ಲ ನಿಮಗೆ information ಸಿಗುತ್ತೆ. In fact, some of you can write an assignment on this, spike proteins of coronavirus. Also spike proteins of different viruses. Influenza virus ಅಲ್ಲಿ ಹೇಗಿದೆ, HIV ನಲ್ಲಿ ಹೇಗಿದೆ, ಎಲ್ಲದರದ್ದು ಬಗ್ಗೆನೂ ಬೇಕಾದ್ರೆ ಒಂದು ಅಸೈನ್ಮೆಂಟ್ ಬರೆಯಿರಿ. It will be interesting. And the last one is pathogenicity islands. ಇದ್ರ ಬಗ್ಗೆ ಏನು ಹೇಳಿಲ್ವಲ್ಲ ಅಂತ ಯೋಚನೆ ಮಾಡಬೇಡಿ. ಇದು ಈ ಸಿಲೆಬಸ್ ಅಲ್ಲಿ ಮೆನ್ಷನ್ ಮಾಡದೇ ಇರುವ ಒಂದು ಟರ್ಮ್, but this is something about the genome of the pathogen which contains areas called pathogenicity islands. So ಇದು pathogens ಅಲ್ಲಿ ಇರುವ DNA ನಲ್ಲಿರುವ ಒಂದು region. So find out what it is, pathogenicity islands. Okay. So with that, I would like to end this presentation. See you in the next class. Thank you.